We're going to talk about a DNA sibling study and what are the things that you can use by studying not only your own siblings, but sets of siblings in your DNA match list. You know, what kind of information can that give you to help you with your genealogical brick walls? And sometimes understanding how siblings inherit from parents and grandparents will help you understand your match list differences, your ethnicity differences. So we're going to go through those things today. So what's the goal? It's to understand our matches better. It's to find other information based on our siblings and to figure out those unknown cousins. We all have those unknown cousins in our match list. So what are full siblings? My mom and dad would probably say partners in crime, but approximately DNA-wise, we are 50% mom, 50% dad, approximately 25% of each grandparent. We all know, according to the Shared Centimorgan Project, that varies. Part of the reason for that is recombination and some other things that can happen, but it does vary a little bit. And each sibling can get a different mixture of mom and dad. Each sibling can get a different mixture of each of the four grandparents. Siblings do have quite a bit of DNA overlap, at least full siblings do. So centimorgans in common, I've listed here my siblings with me and my children who are all full siblings. So I share about 2,800 with my sister, about 2,600 with my brother, and my brother and sister share about 2,600. Pretty normal for full siblings to have that range. My daughter shares 2,800 with one brother and 2,800, give or take, with her other brother. And the two brothers share 3,884. And you might look at that and wonder, why is that so high? Well, that's because Corey and Casey are identical twins. So they share as if they're matching themselves. So that's a full complement of DNA. They're identical twins. So how does this help you? Well, your siblings have DNA that matches to different cousins than you do sometimes. So here's the process that I use to build a chart. And we're going to start with this, and then we're going to talk a little bit more about the inheritance patterns, why you might want to do this, but I want to show you up front how it can be really useful. So if you choose a match to you, so I've chosen a match that's in my list with the initials KA, and I choose shared matches. So that's the shared matches that I have with KA. I've also chosen that same match, KA, to my sister, and then chose shared matches. And then in my brother's list, his matches to KA and shared matches, and so on. So what you want to do is build a chart. Now in my chart, I'm using initials for anonymity because I don't have permission to share all of these names and identities, but you should use real names in your chart. You might want to consider using centimorgan values. There's a lot of other things that you can put in the chart. And capture a PDF of the matches. So the match from myself to KA, I have a PDF. From my sister to KA, I have another PDF of the match list. And from my brother to KA, I have another PDF of the match list. The reason for that is that people sometimes delete their DNA kits or remove their DNA kits. So it's important to archive that information somewhere so that you can go back and look at it later. So this is the list. It's probably kind of tough to see, a little bit small font there, but you can see that my brother and sister and I have quite a few shared matches in yellow. So going down the list, column C is the matches that I have to KA. You'll notice column D is my sister and column E is my brother. So my sister has 8 additional matches in column D in green. Some of those are shared with my brother, but I don't have those matches. And my brother has an additional 9 matches that are in blue that my sister and I don't have. Because full siblings share the same parents and grandparents, These matches to my siblings are assumed matches to our common ancestors. So I can work this complete set of matches as if they were all my own. So think about that. I have 17 more matches than what I had just myself with KA. So this can be really valuable to help you build out those trees and figure out who someone is on your match list. I want to give you one more example before we talk about some of the other things that are going on in DNA land. How does this help you with a set of cousins who are siblings? A set of siblings who are your cousins can have DNA matches to different cousins than you do. So this set of cousins, I've used as an example before, they're kind of a unique set of cousins to me. They're first cousins to me because my dad and their mom are siblings, but they're also more remote cousins to me because my mom and their dad are third cousins. And that third cousin link has not been proven yet. So I'm postulating that my mom and their dad are third cousins. I think they could be in the third to fourth cousin range. So let's look at their matches. So what I did was I looked at the matches that I have with my cousin Bill and myself. And you can see that we had 4 matches in common other than the immediate family that we already knew about. My parents, my children, and so, and his brother and sister, or I'm sorry, his 2 sisters. So we have 4 cousins that match Bill and myself. We also have an additional 12 matches. So once his sister Cindy tested, we had 12 additional matches to investigate. And then Kathy tested later, so we didn't receive any further matches from her. But note that Bill and Kathy have no overlap in the third cousin matches to me. Bill matches me on a set, and Kathy matches me on a different set. It's very interesting, isn't it, how different siblings inherit different DNA from our ancestors? Because remember, I'm only half my mom and half my dad. I'm missing half my mom and half my dad. And my siblings might have picked up other pieces of that I didn't. So they're going to have matches that are going to fill in those gaps. This is particularly helpful because this is a family line where I'm working on an unknown grandparent. And so this is of particular interest to me to have as many matches to investigate as I can. So this shows the DNA matches with Cindy. There are 7 known family and 2 the same as Bill, and 12 additional cousins that we didn't have. So we tried the other sister, Kathy, and she has 7 of the known family members. She doesn't share any third cousins extra with Bill. She has 9 that match Cindy and not Bill, and there are no new matches to search. So sometimes having that third sibling doesn't yield any more information, but you found in the case of me and my 2 siblings, it did yield a lot more information. My brother matched a lot more people than I anticipated. So let's review DNA inheritance a little bit so that we can think about the ways that siblings fit into this. So how are you a match with someone else? And this may or may not be review for you depending on your length of time in genetic genealogy, but there are other presentations on YouTube about this if you're interested in watching. But autosomal DNA, you either are a match with someone because you have whole chromosomes in common with them, or more than likely you have segments of chromosomes in common with them that are measured in centimorgans. For mitochondrial DNA, you're a match with someone else because you have a partial match on mitochondrial DNA, or a full match, which is 0 genetic distance. And the partial match might be a genetic distance of 1 or 2 or 3. So it's a very, very close match, but it's 1 mutation or 2 or 3 mutations different. Same is true for Y DNA. You can have a partial match to someone or a full match with 0 genetic distance. And we've done many talks about mitochondrial and Y-DNA. We are going to talk about that in relationship to siblings, but those are topics that we'll get into a little bit in depth in other presentations. So for autosomal DNA, why do sets of siblings have different matches? Remember that you're a subset of your parents. So you have about 50% of dad and about 50% of mom, and your siblings have potentially a different or overlap with some different 50% of mom and 50% of dad. There's an autosomal pattern of inheritance. Autosomal DNA recombines with creation of every egg and sperm. There's a frequent mutation rate. That recombination of chromosomes gives us randomness and diversity. When an egg and sperm are produced in meiosis, recombination happens when a piece of the material chromosome with the maternal chromosome exchanges at a crossover point with the same piece of the paternal chromosome. This creates a variation in egg and sperm, and those are called gametes. So there's your biology lesson for today. There's more information about autosomal patterns of inheritance. If you're interested, FGS, the Federation of Genealogical Societies, has an upcoming conference in September, and I've recorded an entire session on patterns of inheritance. For autosomal DNA inheritance, this shows visually what one pair of chromosomes looks like, and each pair of chromosomes has possible recombination, and different chromosomes recombine at different rates. For xDNA, remember there's a different inheritance pattern for xDNA. So both men and women have xDNA, but remember, men have one X, women have two. So from dad to daughter, dad's X chromosome passes down to daughter 100% intact. You can see that in the purple here. There's no X chromosome that passes from dad to son. Remember, from father to son, what passes is a Y chromosome. These diagrams are from Jared Smith. You can Google search that and find more information, some really informative blog posts of interest on X pattern of inheritance. The X chromosome passes from mom to daughter and mom to son, but it's usually a combination of both of mom's X chromosomes. So a random amount, somewhere between 0 and 100% of each X is passed. It's not likely a 50-50 split. Normally there's multiple recombinations on the X, and so part of each chromosome that mom contributes to the child is a mixture. So mom passes an X to her son, and mom passes an X to her daughter. So the son has an X that's likely a combination from mom and a Y from dad, and the daughter has an X that's likely a combination from mom and intact from dad. It's also possible for two brothers to be a 0% match on the X chromosome. Each may have inherited different recombined pieces of the X from their mother. So recombination occurred, but they have oppositely recombined pieces. And obviously this doesn't happen very often, but it is a possibility that you have two brothers of the same mother with no xDNA inheritance. So what does xDNA mean for siblings? Let's review. From mother to son, there's one full X that from mom that's likely a recombination of both of her X's, or a copy of one X or a copy of the other X. That's what mother gives to son. Mother gives to daughter one full X from mom that's likely a recombination of both of her X's or a copy of one or the other. And then from dad, she receives one X intact. So two brothers can have the same xDNA, but more likely different xDNA, usually with some overlap. Two sisters of the same father will have the exact same X from dad. It's the only one that he can pass on. So if you have two sisters who you believe are from the same biological father, X DNA will confirm that without any further evidence. And the second X can be the same or likely different combination of mom's X's. So just a note about GEDmatch, I'm going to show you some screenshots from a tool called GEDmatch. I'm not endorsing its use at this time, and we've talked about this in past meetings, but they've had a security breach in July, and so I'm not recommending using them at this time, but I am going to show you some screenshots from there. So this is an X-DNA comparison matrix that you can get if you're doing kit comparison in GEDmatch. I've covered up the kit numbers, even though I've deleted those kit numbers because I'm not sure if they're reusing them or not. I don't think they are, but just in case, I went ahead and redacted those kit numbers. So my mom and dad have no centimorgans in common, so that's a good thing. So if you look at how April and John compare on the X-DNA, if my mom and my dad had X-DNA in common, that would be a little bit alarming. My son Casey and I are a full match. He received one X or the other X or a combination of my two X's. So the testing is reading that as a complete match. And the 196 is the length of chromosome in GEDmatch. 196.1 or roughly 196. Remember, sometimes there's funny end behavior, so these are approximate measures. My son received no X DNA from his dad. Remember, he got a Y DNA from his dad. So my son and I share 196 centimorgans, and that's considered a full, complete match. My daughter and I are a full match. She received one X or the other, or a combination of my two X's from me, and my daughter received one X intact from her dad. She and I share 195 centimorgans, and you might say, well, why isn't it 196? Again, there's sometimes some funny end behavior and other things that account for that. So let's look at a comparison between myself and my parents. So my father John and I are a full match. Again, I received one X intact from my dad. My dad and I share 196 centimorgans. My mother and I are a full match. I received an X or one X or another X or a combination of the two X's from my mom. So she and I share 196. Let's look at the grandparent relationship, because this is where you're likely to see the incomplete matches or the not full matches. So my father John and my son Casey, so grandfather, grandson, share 26 centimorgans. My father and my daughter share 144. My mother and my son share 171, and my mother and my daughter share 56. When a man compares to a man, there is one X being compared to one X in GEDmatch or in any chromosome browser. And so this is my father compared to my son, and you can see the matching segments greater than 7 centimorgans in blue. So that blue line shows the place on the 23rd chromosome, or the X chromosome, where my son and my father share a match. When a man's compared to a woman, his one X is compared to both of her X's. So you'll see here, comparing me to my son, that blue line goes the entire length of the chromosome, because my son and I share an entire X in common. He either has one of my X's, the other of my X's, or the testing company is weaving back and forth between the two to determine that he's a complete match to me. When a woman is compared to a woman, her two X's are compared to the other two X's. So this is a comparison between myself and my daughter. And again, complete match end to end, where we match each other. That blue line means that the entire chromosome we have in common. And if you're not familiar with GEDmatch or reading these kinds of things, there are plenty of free YouTube videos, and there are lots of things at DNA Central about GEDmatch, and many other places you can find webinars. We do want to be careful of the X because the X has small segments sometimes that are considered to be a match, and those small segments can be misinterpreted. So in females, the testing companies weaving back and forth between the two X's, and so the trick to avoid identical by state or IBS is to set the threshold for matching at least at 15. I usually don't mess with anything below 20 or even 25 centimorgans. And I also use sister phasing or mother phasing. We'll talk briefly about that. That's a more involved topic, but I do want to mention it so that you might springboard from there. And we are going to talk about IBS and IBD. So the identical by state and identical by descent are two things we're going to talk about tonight. So for phasing, you can use phasing on autosomal DNA and xDNA. It's most common to use phasing in GEDmatch and other tools like chromosome browsers, but phasing is the process of attributing DNA from mom and dad, or both, to a specific spot on the chromosome. And it helps us map segments on a chromosome, and it tells us that mom, dad, both, or neither is the contributor. And phasing helps us determine whether false positive matches exist. Because if there is a match to me on a company tool site called on Ancestry that's maybe 7 or 6 or 5 centimorgans, if that match is not also a match to my mom or also a match to my dad, it's a false segment. So I can use phasing to determine whether or not some of those matches are real, or some of those matches are what we call identical by state, not identical by descent. So there's a specific way the phasing algorithm works, and this is a lot of verbiage that if you want to, you can go back and pause the recording or read it. But phasing helps us determine which base came from which parent. And if you have mom's, dad's, and child's kit, that's trio phasing, that's the best. But also if you have one parent, you can phase with yourself, and you're at a 94% accuracy with that. So how does the phasing algorithm work? When you download your raw DNA file, this is just a brief little look at what that file looks like. It's just a raw data file. And this file goes from chromosome 1 to chromosome 23, which is your X chromosome. And this file tells you the position and the chromosome number, and also what is on each base. What allele is on each chromosome that tells you mom or dad. It would be nice if allele 1s were all mom and allele 2s were all dad, but that's not the way it works. So if you haven't opened your raw DNA file to look at it, it's pretty fascinating, and it's enormous. It's thousands and thousands of base pairs, so don't hit print. But there's no way to tell which allele came from which parent without segment analysis or phasing or some of the other tools that we use. So what came from mom and what came from dad? You can see that the testing company might read different bases, and we don't know which one came from mom and which came from dad. So if this is mom's genome and this is dad's genome, you can see how it's weaving back and forth between the chromosomes. So let's look at phasing in terms of GEDmatch just for a minute, because this is possible to do if you use that tool. Paternal phased files have a tag of P. So you can see the lower one here in the box. PA79 with my kit number, and P1 is my paternal kit. And my maternal phased kit has a tag of M. And this is the result of my test compared to both of my parents. And the kit number is mine because I'm the base one that's being compared. So what the testing tool has done at GEDmatch is it has read my raw DNA file, compared that to both my mother and my father, and made a half kit of me that matches mom and a half kit of me that matches dad. And then those two kits can be used in GEDmatch's tools. So I can use my maternal kit and find my maternal matches in GEDmatch. I can use my paternal kit and find my paternal matches in GEDmatch. And also I can make phased kits from my siblings. So I can make 2 Todd kits and 2 Jody kits, and that gives me even more complete information. There are some special cases. If you have one parent's DNA and not both, you can really get close to 40% of the information, maybe not half of the information that you received from that parent, but it's close. Endogamy is always a problem. We talk about the problem of endogamy in almost every talk we do. And if you have no parental DNA, you can't use the parent filtering or phasing, but you can use other kits for other kinds of phasing. So you can start with full and half siblings and see if you have sister matches, brother matches. You can test cousins and siblings of cousins. So phasing, there's a lot of information on ISOG, ISOGG, ISOG. Their website has a lot of information about phasing, and you can read about GEDmatch's phasing tools as well. So why would you use phasing? Well, a real match is identical by descent. So that means that it's family, that you actually have inherited a piece of DNA that goes back to a set of common ancestors. The purpose of using phase data for your match list is to improve on your list of matches, so you are eliminating the ones that are false matches. The term IBS means identical by state, which would apply to any false match that randomly happens in a population. It is very common to have identical by state matches that do not go back to a common ancestor, but maybe go back to a biogeographical region. So again, IBS and IBD are something that you can learn more about at ISOG, but I just want to bring this up in relationship to siblings, because you and your siblings at a close level will have common matches, but as you get further and further out, third, fourth, fifth cousins, they might have matches that don't match you, but you want to be wary of small segments. So let's look at a cousin match just real quickly so you can see why the small segments should be avoided. Let's say that you have a cousin with this particular sequence, and you notice right away that that particular segment matches a segment in dad's DNA. So if the segment meets the testing company thresholds of a certain centimorgan value or higher, this sequence would show that cousin 1 matches dad. This is identical by descent, and this ties back to a common ancestor. So that means cousin number 1 and dad are related through an ancestor further back. Let's say you have another cousin match, and it looks like this. Can you see how it's weaving back and forth between mom's and dad's chromosome? So even if this segment meets the testing company threshold, this sequence shows that cousin 2 matches you, but cousin 2 doesn't match mom or dad for that same segment. This is IBS, identical by state, and sometimes it's called a half identical match. So again, you can learn more about IBS and IBD on ISOG, but I just want to show you how the testing companies might weave back and forth, declare someone a match to you. Cousin 2 might be a match to you, a certain amount of segment, maybe 5, 6, 7 centimorgans, but does not match your mom and does not match your dad. That's a false segment. You do not have any DNA that your mom doesn't have or your dad doesn't have. So what does IBS look like in the raw data? It just looks like this, jumping around. And the companies just read the sequence with no ability to tell mom from dad. That's why we have DNA analysis. That's why there's a little bit of elbow grease involved in this. So why should you use phasing? This is from GEDmatch's wiki site. When the longest segment in a match is less than about 12 centimorgans, you need to become increasingly skeptical to the validity of the match. At a segment length of 10 centimorgans, and again, this is not the X, this is just the other 22 pairs, a segment length of 10, there's a 10% chance that that's a random match, a false positive match, or identical by state. The 50% point, where 50% of those matches are false, is between 7 and 8 centimorgans, and the 90% rate at which those results are false segments is the 4 to 5 centimorgan match. So think about what Ancestry is just getting ready to do this month, which is to raise their threshold a little bit. And I think they're raising it to 8 and above, or it might be 7 and above, or something close to that. There's a reason for that, because as you get smaller and smaller segments, you have much higher and higher false matches. So this is kind of a visual of me and my mom and dad and what I've inherited. So to be IBD, or identical by descent, or to matter to us as genealogists, the match must survive phasing. So that IBD match has to match me and my dad, or me and my mom. And you notice that the IBS matches are kind of out there floating around. They don't match mom or dad. So you and your siblings and your mom have the match, or you and your siblings and your dad have the match. So it could be that a match is identical by descent because it matches my brother and my dad, or my sister and my dad. So it doesn't necessarily have to be me, because remember, I had a lot more cousins show up that matched my brother and my sister than matched me. So if it matches me or my brother or my sister along with one of our parents, that segment will lead back to a common ancestor. So what's a close match look like? I want to show you this. This is also through GEDmatch, but this I think is a really important visual when it comes to siblings. So this is the chart on the left is myself compared to my daughter. The chart on the right is myself compared to my son. And remember, my sons are identical twins, so I only need to compare to one of my sons. Now remember, they are full siblings to each other. I'm their mother, and they have the same father. And just as a reminder, there are sometimes when, if you notice, chromosome number 1 has a little gap in there. Look at the first line and the second line. So chromosome 1 starts around 742,000 and it goes to about 247 million. So somewhere around 102 million base pairs, that's where there's a little blip in the test. I don't know why. Sometimes that's something that shows in one sibling and not another. So you notice that my daughter on chromosome number 1 on the left matches me end to end on that chromosome. My son does match me end to end with sort of a little bit of blip in there. And we don't normally worry too much about the blip in there. You notice it's only about 3,000 base pairs or so. It's not very many. So when you look at that, that's a full match on chromosome number 1. Remember, my son got a full chromosome number 1 for me. It's just that the testing company's reading it and there's just a blip in the middle. So sometimes siblings will show differences on their matches, but look at what happens in chromosome 10. Chromosome 10, there's some weirdness right around 90,929,983, and then where it picks up again. And if you notice, there's some weirdness between both siblings, but they both have that weird break in there where the testing company didn't quite read it. And so sometimes siblings show the same anomaly, that there's some kind of blip or something unreadable or who knows. And there's a little bit of oddness in the two, but both siblings have that. And I presume identical twins, the third has that as well. And remember, there's always fuzzy end behavior. So the starts and stop positions can vary a little bit. So let's talk for a minute about Y DNA with siblings. So brothers are almost always going to be an exact match to their biological father. Now you can't say always because there is always a chance for mutation. So fathers and sons will be in the same haplogroup. There's a slight chance of mutation from father to son, but there's a very slow mutation rate on the Y. So if the mutation happens, the father and son will be a genetic distance of 1, and that will show on your results. So this is a picture of the haplotree that goes from left to right in terms of development of haplogroups and migration pattern. If you want to learn more about Y DNA, there's also a video on YouTube that I did about Y DNA. So think about who in your family has the Y DNA, including siblings, that can break your brick wall. So this is the Washer family line, my children's father's family. And so if you notice, there's my first husband, Alan, and his father, Daryl. They would presumably have the same Y DNA. Also, his brothers, Todd, and his one son, his brother, Tommy, no sons, and brother Billy, no sons. So all of those men in the top row have identical Y DNA. Also, going back in time and forward in time, Daryl's father was Thomas. Daryl has a brother, Willard. We talked about Alan and his 3 brothers. My 2 sons, Corey and Casey, and my grandson, Mateo. All of them have the same Y DNA. So think about, in your situation, who are the siblings? Many times we get stuck on our pedigree chart and our direct ancestor lines, and we forget that all of those brothers and all of those uncles and all of those nephews can potentially be key to us. So if it helps you to make a visual display or show pictures or think about how far back can you go to find that Y DNA pattern and who to test moving forward in time, make sure you use pictures if you need it. You can also make an ancestor chart that includes descendants moving forward. So descendancy research is important. You can make that photo array. You can print your tree and highlight where those Y DNA contributors are. There are fan charts. You can use DNA Painter to make fan charts. You can make fan charts by hand. So lots of different ways you can determine which sets of siblings or brothers might be able to help you with your Y DNA dilemmas. So let's talk about mitochondrial DNA for a minute. Full siblings, sisters and brothers, are almost always going to be an exact match to their biological mother. But again, I have to say almost always because we have to leave chance for mutation. Mothers and children will be in the same haplogroup. And there's a slight chance of mutation from mother to child, but again, a very, very slow mutation rate. So if a mutation happens, the mother and child might be a genetic distance of 1. And that, again, is not very common, but it does happen. And I have the haplotree here that goes left to right, similar to the Y haplotree in terms of when those haplogroups were developed in time and the migration pattern associated. So who has the same mitochondrial DNA as me? So again, I make a photo array. So myself and my 3 children. It's a common misconception that mitochondrial DNA is only female DNA. It's not. My sons have mitochondrial DNA too, but they just don't pass it on to their children. Children receive mitochondrial DNA from their mother in the egg, Sperm does have mitochondrial DNA, but it's pretty burned up by the time the sperm reaches the egg and accomplishes its task. So women pass down mitochondrial DNA from their children. So myself and my 3 children all have the same mitochondrial DNA. It's the same as my mother and my mother's sister's children. It's the same as my grandmother Ada and Ada's sister's children. And it's the same as my great-grandma Cora, and she did not have sisters, and so on. My great-great-grandmother Deborah, going on and on and on back in time. We all have the same mitochondrial DNA. So do sets of siblings. My sister's kids, me and my siblings, my mom and her siblings. A lot more people in your family tree might have the same mitochondrial DNA, because that can be men and women. So who has the mitochondrial DNA to break your brick wall? Is there a set of siblings that might have the mitochondrial DNA that you're looking for to determine some information in your family? So you can make an ancestor chart, do descendancy research also moving forward in time, make that photo array, print your tree and highlight. You can use fan charts by hand or on DNA Painter. So there are lots of tools that you can use to determine who has that mitochondrial DNA to help you. So siblings have the same haplogroup, whether that's Y DNA or mitochondrial DNA. So this is my mitochondrial migration map for my mitochondrial group at Living DNA. So Living DNA is one of the 5 major testing companies. So I try as much as I can to use different testing companies at different times. And so I'll show you also, I believe, some from 23andMe. But you can assume that your siblings of the same mother have the same mitochondrial haplogroup and the same migration pattern. This is the Living DNA phylogenetic tree, and I apologize that it's really tough to read, but it looks like any phylogenetic tree in Y DNA or mitochondrial, and it's just a continual series of branching until you get to the point where it has the group where you belong. And you can assume that your siblings of the same mother have the same mitochondrial phylogenetic tree. This is a company called SNP Tracker. And I have used this to determine my husband's Y DNA migration. And so what I did was I put in his haplogroup according to his Y DNA test and his SNP testing. And his haplogroup is I-Y5497. And that's a subset of the IM253 group. But you can assume that any of the siblings of the same father have the same Y haplogroup and the same migration pattern. So that's a really helpful tool to use with siblings. What about some health information and traits? Maybe you haven't considered this, but you and your siblings may or may not have inherited similar health conditions and similar traits. But don't forget, your test results are yours alone. So your siblings will likely have different results unless you're an identical twin or identical triplet. Full siblings, again, share half of mom, half of dad, but likely different segments with some overlap. So if you look at my brother and my sister and I share 2844 centimorgans. This is through Ancestry. My brother and I share about 2600 centimorgans, and we have some overlap and we have some non-overlap. So when you think about health conditions and traits and characteristics, it's important to remember that your health report is not necessarily their health report. Your siblings may or may not have the same health report that you do. There likely is some overlap. You can have a disease and they don't, and you can be a carrier when they're not. So it's just important to remember that when you're sharing this kind of information with your siblings. This is a report from Prometheus back in the day, 2016. I know it seems like forever ago, right? But 2016, Prometheus had an option of uploading your raw DNA from Ancestry or one of the other companies for $5 and getting a health report. Now, this is a pretty no-frills one, and the reason why I've included it, Prometheus has greatly improved their site. It has a great user interface. They were recently purchased by MyHeritage, so there's a lot of great information there. But I just wanted to show you this because we're not going to spend a lot of time talking about the DNA health reports, but I want to show you, so are these things true for my siblings? So I've highlighted a few things. Stronger cravings for alcohol. My family has a family history of alcoholism. Also, significant risk for substance use disorders. Absolutely true. I don't drink alcohol. I haven't for probably 35 years, maybe shorter than that. I don't know. I have to do the math. But a long time. I've not really had alcohol because I know that that's a family tendency. Also, a 1.16 times increased risk of osteoarthritis. That is definitely true for me. I've had arthritis in my hands and my feet since I was 16. So that is definitely a strong family trait. My grandfather had that as well. And a 5 times higher risk for Fuchs corneal dystrophy. And I do have Fuchs corneal dystrophy. I didn't know that I did until I was at the doctor a few years ago, and it was a little bit before 2016 because once I got these results back, I went back to my eye doctor. She didn't think that it was an inherited trait, and I went back to her and said, well, it's in my DNA health report. So there's something to that position. But you'll notice here, the health report tells you which chromosome is associated, which location and which chromosome, the position, the genes, and that sort of thing. So you can do further research if you want. So those alcohol cravings are on 6, substance abuse disorders on 1, so there's multiple places for that. Osteoarthritis is on 7, and Fuchs corneal dystrophy is on chromosome 18. And we're going to come back to chromosome 18 because I'm going to show you something else. So are these things all true for my siblings? I don't know. I would say likely, knowing them as well as I do and their health history, but I don't know for certain. So let's look at chromosome 18, myself compared to my brother and my sister. So why did I look at chromosome 18? Well, that's where Fuchs corneal dystrophy is. And so I wanted to see how much overlap do my brother and sister and I have on chromosome 18. So if you look at the first blue line, solid blue line, that's chromosome 18 myself compared to my brother. So starting a little bit after the beginning of the chromosome, somewhere around 6.5 million base pairs, all the way to the end, my brother and I are a match on chromosome number 18. My sister and I are a match for almost that same amount. And then my brother and sister are a match towards the beginning of the chromosome all the way almost to the end. So we have a pretty significant amount of overlap on chromosome number 18. Now this particular image diagram right here can probably explain the reason why Ancestry states that they'll never have a chromosome browser. Because what I'm able to surmise looking at this and the overlap between my brother and my sister is that if I have Fuchs corneal dystrophy at a certain location, it is quite possible that they will have Fuchs corneal dystrophy if we match our segments at that same location. And this is why Ancestry is saying that they do not ever want to have a chromosome browser, because we do have a ton of information about health conditions and traits that we can intuit from looking at our matches and the match list, not just our siblings, but our cousins, our third cousins, our fourth cousins. we can surmise a lot from looking at that. And I've actually done some of that research. I have a group of cousins, I think there are 18 of us now, who are all descendants of the 5th great grandparent and actually all share a piece of chromosome 15. And so I've been looking at what are the common health conditions that are in that location on chromosome 15. But again, that's a lot of private information that maybe they don't want me to be looking at. So that's part of the reason why Ancestry says they'll never have a chromosome browser. If you look this is the National Institute of Health. They have all kinds of chromosome maps, but this is where Fuchs corneal dystrophy is located, somewhere around 55,543,000 base pairs is where Fuchs corneal dystrophy is located in terms of the approximate location. And you can go through and look up all different kinds of health conditions you have at the National Institute of Health. All of this research is available, it's public, it's transparent. Remember, we paid for all of this with the Human Genome Project. So all of this is available to each of us as a taxpayer. So let's talk about SNPpedia. I just want to mention this really quickly because I think SNPpedia is kind of a fascinating place to roam around and learn some things. You can see where siblings might share some traits. So I just looked up redhead. My family is full of redheads. My brother's a redhead. My father and mother, when they were younger, were redheads. My children are all redheads. So there's a lot of redheads in our family. My grandmother, my great-grandmother, both sides of the family. So red hair is one of those things that is a multiple allele. So there's multiple locations. But this is what I found just from looking up redheaded SNPs in SNPpedia. And you can have red hair for a number of different reasons. And of course, we all know there's a variety of red hair in terms of hue and shade. And so this is red hair and poor tanning ability, and it gives you a link to a study. And I can definitely relate to this. I do not tan. I go from red to white to red to white. There is no tan in this girl. Also, red hair and the responsiveness to anesthetics. Now, I will tell you that I have some very strange things that happen, and I've even been told by physicians that it's a redheaded syndrome, to have a very, very, very high pain tolerance, and then to also have odd reactions or need lots and lots of pain medication to make any difference at all. It's like a high threshold. Also, there's red hair with Irish. Now, I do not have Irish in my background. Most of my red hair comes from my Scottish and Welsh ancestry. I don't have any Irish ancestry, but we all know that there's a little bit of intermingling there of all the different UK populations. There are some blogs here, if you want to read about the prevalence worldwide, if you want to read about the myths of red hair genetics, there's a lot of great information on SNPpedia. So I just wanted to show you that because that's some kind of cool information. Ethnicity charts, let's talk about for a few minutes. Just as your shared matches of your full siblings can be assumed to be yours, the same is true of ethnicity. So let's look at ethnicity estimates and let's look at additional communities. Because whatever ethnicity estimates your siblings have, or a set of cousins that are all siblings have, or whatever additional communities they can add to your genetic communities, you will be able to merge all of that almost into one big super sibling ethnicity and to look at all those different areas. So I'm going to show you some examples of that because I've used this to actually break a couple of brick walls in my family. So let's look at the first set of siblings, which is my children. So I'm going to show you the ethnicity estimates, and I have a chart at the end. So this is for my daughter KYLIE, and you can see the percentages there, and also the additional communities that are there on her DNA health report. I'm sorry, her DNA ethnicity report. If you notice here, my son Casey And even my son Corey, who are identical twins, aren't quite identical in their ethnicity. Not at all surprising. Ethnicity is based on a number of different factors. It's based on reference populations, it's based on population genetics, and it's also based on sampling. So that just means that my one twin and the other twin had slightly different sampling of their DNA for the ethnicity report. So let's look at the chart, because the chart will kind of give you a summary of which sibling and which regions. Now, what you probably notice right away is that the vast majority of their ethnicity is England, Wales, Northwestern Europe. And the next largest category is Ireland and Scotland. Not at all surprising. England, Ireland, Scotland, Wales. That would be what I would expect. We do have a German line, we have a Dutch line. So there is, towards the end of the chart, the Germanic Europe. We also have Norway and Sweden, little bits of Norway and Sweden. But you'll notice that even with the twins, they have different percentages from different regions. But the conglomerate of the three of them gives me far more information than one of them alone. If I wouldn't have had Casey and Kylie, if I would have just had my son Corey, I wouldn't have known anything about Norway. So I wouldn't have known to investigate that or to look at that. And remember, 2% of your DNA or 3 or 4% of your DNA from one region doesn't necessarily mean a whole lot, but it is something to think about. But I'm going to spend most of my time in this England, Ireland, Scotland, Wales region. Let's look at my siblings in the DNA chart for ethnicity. So this is my sister, and you notice that she has England, Wales, Northwestern Europe. She has Ireland, Scotland, a little bit of German and Sweden, and the communities that she has in US. My brother has a couple of communities that show up that aren't in ours. And again, he has similar ethnicity results. I'll show you the chart in a minute. And mine. But there are some communities that my brother and my sister have that I don't have, particularly that New Jersey one. And I know the New Jersey one for my brother is Dutch. We have a line that comes from in the mid 1600s into New York and migrates into New Jersey. So that must be represented primarily in my brother, but I can assume again that anything that shows up between the ethnicities of myself, my brother, and my sister, I can attribute all to me because we are of the same two parents. So again, you can look at this chart and you can see that England, Wales, Northwest Europe is one big chunk, and the next big chunk is the Ireland, Scotland. And again, ours is almost all Scottish, at least that I've proven on paper. We don't really have Irish that I know of, and I always leave room for that because I don't know all of it, but we have some lines that go back to the 1600s so far in Scotland. Germanic Europe, my sister has quite a bit more than the rest of us. So if I would have just looked at mine, I would have thought, gee, that's a low amount of DNA for Germanic Europe, 2% for me, but my sister has much higher representation there. And for Sweden, again, I have Swedish, my sister does, but my brother does not. And then for Norway, my brother and I have it, my sister does not. So the conglomerate of the three of us gives us much more information. And let's look at one more set of siblings, those three siblings that I've been talking about today, my three first cousins, Bill, Kathy, and Cindy. If you look at theirs, what you'll see is the vast majority, again, they're my first cousins. So England, Wales, Northwest Europe, Ireland, Scotland, all as predicted. Germanic Europe, they have much higher percentages there. Sweden, my cousin Bill doesn't have any Swedish represented, but Catherine and Cindy do. And they all have Eastern Europe and Russian. Russian. And this is something that's been really important in our family because we've been trying to break the brick wall of their unknown grandfather. And knowing that there's Eastern European and Russian has allowed us to narrow down the unknown grandfather to 5 Russian men who lived in the same coal mining town as grandma. And so we've been able to really narrow down to 5 men at this point in that one small, teeny tiny town. And they had come there, those Russian men, to work in the coal mines. And so that's really given us a leg up on that research. It's not definitive, but it definitely has helped point us in a direction. So sets of siblings just might hold the key to your brick wall. Not just your siblings, sets of cousins who are siblings. Maybe you're working on your husband's line, his siblings. Use those sets of siblings to your advantage. Best of luck.